Hi guys, today we're going to be talking about the cell cycle and mitosis, and let's start with a silly joke. Biology is the only subject in which multiplication is the same thing as division. Hopefully you get it. It's a little nerdy. Alright, so let's talk about the cell cycle. You may have heard this only referred to as mitosis before, but really the cell cycle is the entire life of the cell. And if you look at this diagram over here, and it has all these pieces, and then just this one little section of it is actually mitosis. So we need to be careful about when we're talking about the cell cycle, which is all parts of the cell's life, versus just mitosis, which is just the division of the nucleus. So we'll keep talking about that. Alright, so you started out as a cell smaller than the period at the end of a sentence, and now you are big and fabulous. These are pictures of some of Miss Fogler's former students. I don't know you all yet, so I can't have pictures of you. But we'll get there. Okay, so how did you get from a single cell to a baby? Your original fertilized egg had to divide, and then divide again, and then divide again, and again, and again, and again, and millions and millions and millions of times. So why do our cells divide? Well, there's many reasons. First of all, they're going to divide. Some cells are going to divide as their only form of reproduction. So single-celled organisms, the only way they divide, or the only way they reproduce is through mitosis and this whole cell division. Okay, so one-celled organisms are doing asexual reproduction. In your body, you divide for growth. You're not bigger because all of your cells have gotten bigger. You are bigger now because you have billions more cells than you did when you were first born. Okay? And then also you're going to need new cells to repair and replace cells that you've lost based on just living. Okay? For example, every time you just rub your arm, you're sloughing off dead skin cells. So you need to divide and replace and grow more skin cells. The inside of your digestive system, your stomach lining, is constantly being worn down by the high pH in your stomach, and then your body is making new cells to replace all of that. Alright, so remember, going back to some cell structure and function, let's talk about some of the organelles that are going to be involved in the cell cycle. Okay, so you're going to have the nucleus, because what does every new cell have to have a copy of? All of the DNA. Okay, so every new cell needs a full set of chromosomes, all the DNA. Remember, DNA is what allows your cells to work. So every cell needs a full set of chromosomes. Then also your cytoskeleton. Remember, your cytoskeleton helps keep your organelles in place and helps move them from one location to another. So that's going to be really important as we move things around. And there's going to be specific parts of your cytoskeleton, like your centrioles in animal cells, and then microtubules and spindle fibers that will be really important for moving things around. Alright, so just some reminders about the nucleus. Remember that it's protecting the DNA, it's got an envelope around it, and that envelope has pores in it, okay, to allow some things in and out. What kinds of things we need to get in and out? Remember, you need some large macromolecules to be able to get through, specifically RNA. Okay, remember that DNA carries the copies for making all the proteins that you need in your world, but the DNA never leaves the nucleus. So RNA makes a copy, heads out, goes to the ribosome, makes a protein. Okay, so here's a picture of the nuclear membrane with a little pore in it. What is the function of your cytoskeleton? Remember, it's to provide support for your cells, keep them going, and then also to anchor the organelles and to move them around. Also, it's going to help the cell move if the cell needs to move. And it's going to help regulate what's going on. So your cytoskeleton is this whole huge framework of fibers and tubes and kind of tracks that things move from one direction and the other on. Your organelles are kind of cemented into that. Okay? One of the specific parts of the cytoskeleton that is really important for cell division are your centrioles. And centrioles exist in animal cells. Okay, and they are going to organize microtubules, the little bitty tubes that are going to grow out and help move the chromosomes around. So you can see in this picture, here are chromosomes, and here are the microtubules that are going to grow and kind of work like fishing line almost, directing traffic of those chromosomes. Okay, all right, so what is going to be passed on from an original cell down to its daughter cells? Well, the daughter cells are going to have an exact copy of the genetic material. The DNA inside a cell is what makes it able to do the things that it does. So if it doesn't have DNA, it's not going to work. Okay? And mitosis is the portion of the cell cycle, all of actual division, that is dividing up the DNA. Okay? Okay? Then each cell is also going to need organelles and cytoplasm and a membrane and enzymes and all those things. And cytokinesis is the portion of division that allows for the separation of those things. Okay? All right, so let's look at our little overview of mitosis. Okay, so here's what the various phases of mitosis are going to look like. Here's interphase, and the cell just looks a lot like a cell. And then prophase, prometaphase, metaphase, anaphase, and telophase. You may not have heard of prometaphase before. It's not in any of the freshman biology books. We don't really talk about it, but it's not much different. It's just kind of an in-between between prophase and metaphase. Okay. So, and then people like to try to remember this as IP mat, IP on the mat, to remember the order: interphase, prophase, metaphase, anaphase, telophase, and then there's cytokinesis. I also had a student my very first year teaching come up with a really silly one that I have always been able to remember, and it was, if people marry aliens, that's crazy. So IPMATC, if people marry aliens, that's crazy. Come up with anything that's going to help you remember the order of these, because you just need to remember which one's happening when. Okay, so let's talk about what's happening in each portion. So interphase is the first phase of the cell cycle, and it is the longest phase of the cell cycle. It takes up 90% of the cell's life. And this is when the cell is just being a cell. It's doing its everyday job. It's making proteins, it's making enzymes, it's making digestive enzymes. If it's a stomach cell, it's just doing whatever it does. Okay? It's going to produce RNA. There we go. Okay? And when it's triggered for division, and we'll talk about what some of those triggers are, it's going to start getting ready to divide. Okay? All right. The cell has a life cycle, so that's what we call the cell cycle, is overall the life cycle of the cell. Once a cell is formed, it has a couple different pathways it can go on. It can grow, mature, and divide again. So it's going to go through all the phases of interphase and then mitosis. So the phases of interphase are G1, S, and G2, and then M is mitosis, or the actual division part. Okay? And the types of cells that do that in your body, here's just a few examples. Okay? Some cells are going to grow and mature and never divide again, and they're going to stay in their mature form for the rest of your existence. They're going to live as long as they can, but you will never make more of them. So they go from G1, that first G portion of interphase, to a G0 phase, and in G0 they just stay there. So example of this would be brain and nerve cells and muscle cells. So if you kill brain cells, people, they're gone, and you never get them back. Just a little reminder to make good choices. Some types of cells, like liver cells, can go from a G0 phase and, based on need and necessity, can be activated to go over and start dividing. But it doesn't happen very often. All right, so let's talk about what's happening. So interphase is divided into three phases. G1 is the first gap phase, or the growth phase. So the cell is going to be growing, doing all the things it's supposed to do. Given the right kind of trigger, it's going to go into a G0 phase and just stay there forever. Okay? If the cell ever gets a signal to divide, and we'll talk about what that is in the next lecture, then it's going to move into the S phase, and S is for DNA synthesis. So this is where all of the DNA is going to be replicated. Okay? Then after the S phase, the cell goes into G2, or the second growth, or the second gap. It's going to do a little bit more growing. It's going to prepare for division. It's going to make copies of its organelles, its proteins, its membranes, to make sure there's enough stuff for two cells, and it'll get ready. All right, so what does the cell look like in interphase? Well, the nucleus is well defined, and the DNA is loosely packed in long chromatin fibers, so the DNA basically looks like spaghetti in the middle of the nucleus. Okay? As it's starting to prepare, it's going to replicate those chromosomes, so there'll be even more there. With a regular light microscope, you wouldn't be able to tell the difference between before DNA replication and after. Okay? But also, as the cell is preparing for mitosis, it's going to make more copies of organelles to make sure everything is ready to go. Okay? So we'll talk some more specifically about replication of DNA when we get to the DNA unit second semester. But for now, just remember that the DNA is going to be able to, the two strands of DNA, unwind, and each is used as a template to make a new copy. Okay? And it's really important that the new copy is exactly identical to the original copy. Okay? You've got over 3 meters of DNA in each cell. That's a lot of DNA, okay? And each daughter cell has to get a whole complete copy. The error rate is about 1 per 100 million bases, so mistakes do happen, absolutely. Okay, so it's about 30 errors per cell cycle. Most of those errors aren't going to be a problem. We'll talk tomorrow about why some of them are. Okay. All right, so how is all that DNA, because you have about 3 meters or 9 feet of DNA in each itsy bitsy little cell, so how is that organized so it all fits in? Well, it gets wound up very, very, very tightly. So let's talk about DNA organization. So we call it a chromosome. So remember, DNA is your double helix. And that double helix is going to wrap like a string around a spool around these circular proteins called histones. So here's the second layer of kind of packing is what we call this. You get your string of DNA, your double helix, and it winds around these circles, these circular proteins called histones. Okay. Then those histones are going to get so wound up that they start kind of winding and joining together into big complexes. Okay, and this right here we call chromatin. Okay, and if you zoom out from this, it would look like a really thick fiber, kind of like going from a single thread to going to a thicker shoelace. It's kind of the principle of going from just individual strand of DNA to the thick chromatin, excuse me. Then the chromatin starts winding up on itself and spiraling and kind of folding up on itself, and it gets to this kind of mess and then gets really, really, really tightly packed into a full actual chromosome. Okay? If you've ever sat around and played with a corded phone, you'll notice that while it's a spiral, the more you play with it, the more it starts to fold up on itself and kind of jumble and pack in really, really tight. It's the same kind of idea with DNA. So hopefully that's something that makes sense for you. Okay. So after the DNA has been replicated in the S phase, it's going to start to condense and start to form these really tight double-stranded chromosomes, okay? So one half of this X, this whole side of the X, is one set of the DNA, and then this whole other side of the X is the other set of the DNA, okay? So each half of the X is identical. So this stuff is identical to this stuff, and they're held together in the middle at a spot called the centromere. Okay, so that's what double-stranded human chromosomes look like. Some of them are much more obviously an X. Some of them are kind of lopsided. It depends on the different chromosome and what kind of information is on them. Okay, so a duplicated chromosome is going to be made of 2 sister chromatids, chromatid, held together. They're held together at this middle spot called the centromere. Okay, and the one side of the X is exactly the same as the other side of the X. We also are going to talk a little bit from time to time about homologous chromosomes. Homologous chromosomes are the 2 chromosomes that you have that carry the same types of genes. So remember, you got half your information from your mommy and half your information from your daddy. Okay, so you got 1 copy of a gene for hair color from your mom and 1 copy of that gene from your dad. Well, here's the chromosome that carries the copy from mom. Here's the chromosome that carries the copy from dad. So since these 2 chromosomes have the same genes on them, we call them homologous. Remember, homo means same, logous means information, same information. Okay, so here we have single-stranded chromosomes, and then these are double-stranded homologous chromosomes made of sister chromatids. All right, so now that we've kind of beaten interphase to a dead pulp, let's start talking about mitosis and what's happening during the division of the nucleus. So remember, this is when the nucleus divides, when the DNA is actually separated into 2 new nuclei. Some call it the dance of the chromosomes. Remind me when I get back, I might be able to show you a really fun, silly, or actually if you YouTube it, there's some very funny interpretive dances out there about mitosis. Good nerdy entertainment for you. All right, mitosis itself is divided into 4 phases: prophase, metaphase, anaphase, and telophase. All right, prophase, what's happening here? This is where the chromatin is going to really, really condense and the X's are actually going to become visible. Okay? The centrioles are going to start moving to opposite ends of the cell. Okay, and they're going to start producing the mitotic spindle, which is going to work like the fishing line that's going to move the chromosomes around. Okay? And the nucleolus. Remember, the nucleolus is the dense spot in the middle of the nucleus where ribosomes are made. So that is going to break apart and it won't be visible anymore. And also the envelope, the membrane around the nucleus is going to start to break down. All right, then there's prometaphase, which is the transition to metaphase. And in prometaphase, the spindle fibers actually attach to the centromeres of the chromosomes and actually will then start moving them in locations. Okay? They attach to the centromere and they form the structure called a kinetochore. Okay? Which is just really the site of attachment. Okay? Connecting centromeres to centrioles. And the chromosomes are actually going to start being moved in a specific area. Okay? So prophase, the chromosomes become visible. Prometaphase is when the spindle fiber actually attaches. All right, in metaphase, the chromosomes are going to be aligned along the middle of the cell. Okay, so MM, metaphase, middle. So you'll be able to remember that from the M's. So they get into a nice little line along the middle. The pictures always make it look like it's this really, really neat single file line. You'll get to see some slides of cells later in a couple of days or so, and you'll see that it's really very jumbled. But it's still lined up along the equator of the cell. Okay, we also call that spot where they line up the metaphase plate. Okay, the spindle fibers are what is going to move the chromosomes to that location. And it's going to make it easier so that then they can make sure the chromosomes are divided separately. If I wanted to divide the room in half, it would be easiest to put you all in one spot and then be like, okay, you there, you there, you there, you there, rather than just trying to kind of decide how to divide the room in half when you're all standing anywhere you want. Doesn't make it as efficient. Okay, so lining them up and then pulling them in half is going to be the most efficient way to get the chromosomes evenly divided. All right, so here's another picture of metaphase. Now, anaphase is when the chromosomes actually Anaphase is when the sister chromatids actually start to separate. Okay, the spindle fibers are going to start pulling, and the two sister chromatids held together at the centromere are going to pull apart and actually start being pulled towards the opposite ends of the cell. Okay, and they're going to be pulled by really cool walking motor proteins. So it's literally this little protein that walks along the spindle fiber, and it carries the chromosome with it. Okay. And as that happens, the microtubule is also going to be broken down. Okay, so this takes energy for this walking motor protein to actually move. So you're going to need a lot of ATP to divide your cells. That's why they talk about, oh, when you're in a growth spurt, you sleep so much, because you literally have to make up for all the energy that you've expended growing and doing mitosis. Okay? And the poles of the cell are going to start moving further and further apart because some of the microtubules aren't going to attach to chromosomes. They're going to just be loose, and they're going to keep growing and growing and growing, and as they grow, they're going to keep pushing the other parts of the outer parts of the cell away from each other. Okay. So the two sister chromatids are going to divide in half to become individual sister chromatids or individual chromosomes, and they're going to get pulled to opposite sides. Okay, and again, this happens. Here's your little picture of a motor protein actually walking the chromosome down the microtubule. Okay, and then the microtubule is also going to get shortened down here. So as the protein walks past a section of the microtubule, that microtubule is going to get broken down. So it just kind of disappears and goes away. Well, it doesn't go away, but it goes back into its basic pieces, so it could be built again later. All right, telophase is where you're going to start to get two actual distinct nuclei. Okay, so when the chromosomes arrive at their separate poles, then a new membrane is going to start forming around each collection of chromosomes. Okay, the nucleoli, remember that's the plural of nucleolus, those are going to start to reform. The chromosomes are going to start to unwind and start looking more like spaghetti. Okay, and the spindle fibers are going to disperse, break down. Okay, and cytokinesis is going to begin. You'll very rarely see a picture that tries to separate the moment of telophase from the moment of cytokinesis because they really start happening at the same time. Okay, so cytokinesis is the division of all the other stuff, all the cytoplasm, all the organelles, and actually pinching the membrane in half. The way it works in animals is that you get a whole band of actin microfilaments that start to constrict like a belt around the equator of the cell. We call it a cleavage furrow. Okay, that's a cleavage furrow, and that cleavage furrow is going to work like taking a string around a balloon and tying and tying and tying and tying and making it tighter and tighter and tighter and tighter till the two pieces can be separated from each other. Okay? All right, so here's a nice little nifty animation. It goes pretty fast, but you can see some of the pieces. I'll show you another little animation in a bit. So here is the actual cleavage furrow. You can see how it's being pulled tight to separate the two. Here's some pictures of mitosis in a whitefish cell. So interphase, prophase, metaphase. You can see, I mean, they're in a really distinct line, but it's a really jumbled line of chromosomes. It's not a really neat line. And then here they're starting to be pulled apart. Here they've been really pulled apart. Here's late telophase, where you can actually start to see two distinct nuclei and the cleavage furrow going in the middle. Right, and there's another really pretty picture of it. Isn't that fun? All right, plants have slightly different cytokinesis because they don't just have a membrane. They also have a cell wall. So the cell wall and the membrane have to be formed to make your two distinct cells. Okay, so the way that happens is that a cell plate starts to form in the middle of the growing cell. So once the two nuclei are starting to form, vesicles, remember little bitty membrane-bound packages of something, are going to start to line up at the equator of the cell. So they're going to line up like this. They're going to move together till they fuse and make a whole big cell plate surrounded by one big membrane. Okay, and then they're going to keep more cell walls going to be built and built and built and built and built until they then reach the edges, and these two new cells are now separated. Okay, so here is cytokinesis happening in a plant cell. You can see all these vesicles, little bits of cell with cell wall in them, and they're going to join together to make one big one, and then they're going to keep growing out and out and out and out until they reach the edges, and now your two cells are separated. So there are some nifty pictures of mitosis in a plant cell. And this is what we're going to look at when we look at slides of it. Here's some slides of onion root tip, and that's what you're going to be looking at when you do your lab on mitosis. Okay. Now, remember that mitosis, just like everything else, every other process that we do, evolved from some other earlier process. So binary fission is the way that bacteria reproduce, and bacteria have a single circular chromosome, and they have no membrane-bound organelles. So they just first have to copy that chromosome. Okay, the two chromosomes separate from each other. The ring of proteins happens, and then the cell pinches in two. Okay. So as we've gotten, as our cells have gotten more complex, there's had to be a few more complex situations that really separate us out. So you can kind of see how mitosis looks in some different organisms as we go from bacteria to simple protists to slightly more complicated protists to single-celled eukaryotes to animal cells. So you can kind of see, and we're going to keep talking about evolution all year long, but you can kind of see the progression of going from really, really simple to more complex stuff. All right. Hopefully that makes sense and you'll enjoy your lab. See you guys later. So right now we're going to look at a couple different animations and a video of mitosis and cytokinesis so that you can see what's happening, try to get a visual of what is going on in this whole process.
Mitosis and cytokinesis. Mitosis, division of the nucleus and its chromosomes, is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is followed by cytokinesis, when the cytoplasm splits to form 2 separate daughter cells. The cytoplasm of a cell in late interphase contains 2 centrosomes, each of which may contain a pair of centrioles. In the nucleus, the chromosomes have been replicated during S phase, but are still dispersed in the form of chromatin. 1 or more nucleoli are present. During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of 2 identical sister chromatids joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle. During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole. At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles. Anaphase begins when the two centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, the spindle microtubules not attached to chromosomes lengthen, pushing the two poles farther apart and elongating the cell. During telophase, nuclear envelopes form around the identical sets of chromosomes at the two poles of the cell. The chromosomes uncoil, and nucleoli appear in the two new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the two daughter cells. In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
Indicate the correct order for the stages of mitosis by dragging the numbers over the cells. Click the check answer button when done.
Okay, so hopefully you guys can do this on your own, but I am going to kind of help you out with it a little bit. So first is interphase. Second is prophase. Third is prometaphase. Good. Fourth is going to be metaphase. Then anaphase and telophase. Now, I-P-M-A-T. IP on the mat. And there are applause for you. Isn't that great? Alright guys, so now I want to show you a little video of mitosis and cytokinesis happening in time lapse.
Cell division functions in asexual reproduction and in the growth and repair of organisms. Cell division consists of two processes. Mitosis, the division of the nucleus, is usually followed immediately by cytokinesis, the division of the cytoplasm. This time-lapse video of cell division in a newt lung cell speeds up division by a factor of 100 or so. You will see a process that normally takes more than an hour happen here in less than a minute.
Okay, so I'm going to replay this a couple times without the sound on so I can kind of walk you through what's happening. Alright, so let's look at it again. So there we've got your chromosomes, and you can see them starting to condense. And you can't really tell or see that they're X's. I mean, if you look really closely at that one, it kind of looks like an X for a second. So you'll see, I mean, a lot of that stuff that we talk about is kind of arbitrary. So they're starting to get to the middle, they're lining up. You see how it's not a very neat line, but they are lined up. And then you'll see, look, oh, there's anaphase. They start pulling apart. Okay, you see, as soon as they start getting apart, they start unwinding, and you get just this big mass again, rather than individuals. And then there's the beginning of your cleavage furrow happening. Right there. So you get to the point where you're just about two individual cells. Alright, let's watch it one more time. Alright, so prophase. More prophase, they're moving and getting to the middle, so that's prometaphase. Getting closer and closer to metaphase. And they're all in the middle. Looks like that's about it. Now, oh, there goes, starts anaphase. As soon as they get to the edges, you've got telophase going. and then there's cytokinesis happening. So hopefully you'll notice that really this is just one continuous process, and that we have put labels in at certain points just to make it easier to talk about, but it's not like the cell says, okay, time to do metaphase, everybody line up, everybody line, okay, good, no. The cell doesn't have these exact distinctions, it's names that we put on it, okay? Alrighty, see you guys next time.